Right. So this is a project I did. It's sort of been a project, to be honest, that's been picked up and put down over the years, and we finally gotten it sort of wrapped up and finished. So it's really also nice to talk about this work, for hopefully in front of people who appreciate flies and care about what they're doing. So this is a picture of Morgan back in the fly lab that he started back in 1900 whatever, with his Drosophila melanogaster. And what you can't see over here is a picture of me out in Vienna, in Kallenberg, I think, with the piles of pumice from the vineyards. And if you hit this pumice with a stick, you see a cloud of melanogaster fly up. And that is a really good way to collect lots of flies, as Christian found out, and I later also exploited. You can get almost pure melanogaster with only a few percent simulans in the thousands, in the milliliters. I once collected 50 mils of flies. So what are the differences between these two sets of melanogaster, the ones that Morgan started to work on, the ones I can go out and collect? We're used to thinking of adaptation as you have some SNPs and you have change through time, and that's very true, and that's evolutionary change and particular evolutionary adaptation. But there are other things that are different in this period of time. In the last, well, since Morgan started working, there has been a difference in the Wolbachia that melanogaster harbors. So this is an intracellular bacteria, a symbiont, and it went from one strain to another. There are also new transposable elements that have entered since the time Morgan was working. So there's a lot of evolutionary change that's not, strictly speaking, adaptation due to SNPs and selective sweeps and the kinds of things that we all know and love and are used to, but also adaptation due to these cobionts or transposable elements. So what are transposable elements? Probably many of you already know this, but I give the standard introduction. So transposable elements are bits of DNA that are inherited from parent to offspring, like other genes, but they also replicate within genomes, so they can change their copy number through time. This is a really successful evolutionary strategy that has evolved multiple times and gives rise to a lot of the DNA in genomes. So if we look at the human genome is roughly 45% transposable elements. You compare that to their protein coding genes, they're only about 1.5%. So there's a lot of DNA that's derived from transposable elements in the human genome, most of which are now inactive. So they're no longer jumping around and replicating. Humans aren't particularly special. Lots of genomes have lots of TE-derived DNA, and it's a really good explainer of genome size. The more TE-derived DNA you have, the bigger your genome is. TEs are not just inert junk floating around in genomes. They can also be sources of mutations. So these are some of the famous mutations that were caused by transposable elements. This peppered moth mutation, is the famous story of adaptation. My colleague in Liverpool found that it is, at least the British allele, is due to the insertion of a large transposable element. Mendel's Reekley P mutation is, of course, probably the one studied by Mendel back in the early, in the monastery. That also seems to be due to a transposable element insertion near a gene, and the same for the best mutation of all, the white 1 allele, that is a dock insertion near the white gene in Drosophila melanogaster. So these TEs are producing a lot of different genetic variation. The one I'm going to talk about is one called the P-element. So the P-element was originally discovered, I only learned this recently, when two population geneticists, Chuck Langley and John Gillespie, went on a road trip collecting flies around Texas. They were mostly unsuccessful, but they got to this port near Harlingen, where they collected a net full of flies, and in there was the very first P-type strain that harbored P elements, a new transposable element. I happen to know, because I grew up in that area, that that is where the banana boats come in from Central America. So it is quite likely that that was how the P-element got to Texas. Once, like me, the P-element grew up and went to university. It went to UT Austin, where it was originally studied by Dr. Horizami, who was still there when I was a student there, and then later picked up, the study of it was later picked up by Margaret Kidwell and Bill Ingalls, who did a lot of work characterizing the molecular basis of these strange phenotypes, which I will tell you about now. So why the P-element was discovered was because it had these strong phenotypic effects. So these phenotypic effects were present when you took a wild, recently collected male and crossed it to a lab female, so an old lab strain. You do this cross with the wild Texas male and the old lab strain, and probably you can't see this very well, but what you get is the offspring have all sorts of problems. This problem here is occurring in the females, where they have shredded ovaries. They have ovaries with almost no eggs in them. If you do the cross in the reciprocal direction, everything's fine. So there's something about the direction of the cross that's causing this problem. It turns out that much later it was discovered that what's different, what's wrong, is that you have a set of small RNAs that match the P-element, which are present in the wild flies and absent from the lab flies. Oh, I've swapped the order of these. That's terrible. So when you have these small RNAs present, they suppress the activity of the P-element, and you end up with perfectly fine offspring. If they're absent, then you cannot suppress the P-element, and you end up with active P-elements that shred, that cause double-stranded breaks when they're active. And because they're so active, because there's so many more P-element insertions in these wildflies than in the old lab females, which are completely lack them, you end up with lots and lots of activity of the P-element, and you end up with really severe phenotypes. Okay, so why is the P-element absent from the lab strains? It's because of when they were collected. So they were collected at some point in the past, say in the 1930s, and that was before P-elements existed in wild melanogaster populations. You look many years later, and wild melanogaster populations are full of P-elements. The data used to infer this are the fact that P-elements are absent from close relatives of melanogaster. So that's melanogaster on the left with a big smear. And these are other species that are close to melanogaster, which are completely lacking the smear that it's due to the P-element. I like showing this slide so that I can point out to everybody that back in the old days, you could get a Nature paper from running a gel. No longer true. In Margaret Kidwell's lab, they went out and they found the source of the P-element, which is a Caribbean species called Willistonii. They think it's the source because the P-element that exists in Willistonii is very similar genetically to, one base pair different to the P-element that occurs in melanogaster. And this is despite those two species being 54 million years apart. Right, so this was very surprising that the P-element had jumped, but the P-element didn't stop going. So this is, the P-element jumped from Willistonii to melanogaster, and then it went on from melanogaster to simulans, again accompanied by a single base pair change. So this is very strange. These two species don't hybridize. These two species, in principle, can hybridize, but you need to rescue both lethality and fertility in order to get fertile hybrids. So this is probably not happening just by simple hybridization, but by some unknown mechanism. This is a very, I like this little cartoon. It was drawn by my master's student. I'll point out that she was a bioinformatician, therefore didn't look at flies and maybe never realized they don't have 4 wings. But nevertheless, she's a good artist. Okay, so how we went about studying the P-element in simulans was very initially by just looking at hybrid dysgenesis that was occurring in simulans and then associating it with the presence of P-elements in that species. And it's a very rapid spread of the P-element. I'm trying to rush through quite a bit of stuff because I know this is very familiar to some of you, particularly as we did this work here. But if you look before 2009, you've got lots of beige in these pie charts. The beige represents the proportion of lines collected that lack P-elements, and the red is those that have them. If you look before 2009, you don't see much. If you look after 2009, there's a lot more. Generally, everything you collect after 2012 will have a P-element in it, so it's completely invaded in this species. So when we look for hybrid dysgenesis, we looked in the females. So you just, did this phenotype because it was quite an easy phenotype. You just dissect a bunch of females. My PhD student at the time could dissect 1,000 in a day. So he could score 1,000 females for hybrid dysgenesis every day, which was good because he scored a lot of flies. However, what we didn't look at was what was going on in the males. And so that brings us to this project. We just wanted to know what was happening in the males. How are they experiencing hybrid dysgenesis? So you do this cross in the dysgenic direction and the other direction, and you compare F1 males and ask how their phenotypes are different. Okay. So this is not the first time dysgenesis has been studied in males. In melanogaster, it was actually first originally described in males. So Horizonme originally got interested in this Texas strain that had P-elements because he saw that the F1 males had recombination, which is unusual for Drosophila. Mostly males don't recombine, but these males were recombining. Is probably related to the fact that they were experiencing double-stranded breaks and repairing their genomes by using homologous chromosomes. In Margaret Kidwell's lab, they also did some of this work and showed that they showed some of the same effects as females at 29. So they have some of the atrophic gonads and other phenotypes that you see associated with hybrid dysgenesis, like mutations and chromosomal abnormalities. So we wanted to look at these in simulans and also use some more modern technology to look at them, at these dysgenic phenotypes. This is, as I said, it's been some work that's been picked away at over the years by different people in my lab, but it was largely driven by Valeria, who's here, and Joe, who's there, who did the lab work and the analyses together. Okay, so we compared dysgenic males and reciprocal controls. If you're a smart geneticist, you may see that there's a problem, and that is if you're making reciprocal crosses between strains, your males are not genetically identical, and that's of course because of the sex chromosomes. For females, it's easy. Reciprocal crosses give genetically identical females, and their mothers are different. So the dysgenic females come from M-type mothers that lack P-element piRNAs, and the control females come from P-type mothers that can protect their offspring. If you do reciprocal crosses and get males, they differ in their sex chromosome complements. So the dysgenic males will again come from M-type mothers that lack the P-element piRNAs. So they'll have active P-elements. The control males will come from the P-type mothers that have P-element piRNAs and suppress the P-element. So the controls should be fine. The dysgenic ones should show dysgenic phenotypes. But the dysgenic males get their X from the M-type stock, and the control males get their X from the other stock, from the P-type stock. And this is not a great comparison because there's a lot of dysgenesis that we know from melanogaster populations maps to the X chromosome. So this is not only a genetic difference, but potentially a very important genetic difference. So how do we get comparable dysgenic and non-dysgenic males? So we used something called an attached X. We're not the first people to do this. Lots of people have done this over many years, but it's one of these cute Drosophila tricks that there's a lot of in melanogaster, and there's at least this much in simulans. So attached X crosses work because the females have physically attached X chromosomes. They segregate together. And this means that instead of passing on X chromosomes to their sons, which would be the normal state, they pass on a Y chromosome to their sons. And these sons get their X chromosome from their father. So the inheritance of sex chromosomes is reversed. If you do the reciprocal cross, the males of this attached X cross are perfectly normal, standard XY males, and they pass on a Y chromosome to their sons, just as God intended, and the sons also get X chromosomes from their mothers. So those, these guys have swapped sex chromosome inheritance, and you end up with genetically identical F1 males, but importantly, they differ in who their mothers are. So these guys have mothers that have never seen the P element and that lack suppression, and they'll have active P elements. And these guys have P-type mothers who have seen the P element, have the suppressive small RNAs, and should be normal. Okay, so those are the males we're going to compare. And we did some stuff. We did RNA-seq, we did some transposition assays, and we looked at small RNAs and some fertility assays. So just a bunch of stuff. So the first thing we did was RNA-seq. Some people have suggested that when P-element dysgenesis happens, it unleashes a lot of transposable elements. Other people have said this doesn't happen. All I can say is we don't see it. What we see is we did RNA-seq on dysgenic and control testes, and we found that the dysgenic testes have mildly elevated P-element expression. It's roughly 3-fold higher than the reciprocals, and it's significant, but not wildly. And it's the only TE that we see that's differently expressed between dysgenic and reciprocal males. Okay, not necessarily a surprise. We also compared the dysgenic types to the P-type parent. This is a little bit of an uglier plot. There's a lot more going on. I'm not sure how much of it is interesting, but we looked at reciprocal versus the P-type parent, and you see that the P element is not very highly expressed compared to other TEs. If you do the reciprocal cross, you see that the P element is, it's not the most significant TE, but it's up there. So this suggests that even though the P-type parent has P elements that are expressed, they're even more expressed in the dysgenic type. So there's even more expression than there should be in a suppressed strain. Okay, so the P element has higher expression in these dysgenic flies. And the next thing we asked was, were there any consequences of this dysregulation? So the first thing we did is this transposition assay. So how we did this is we compared transposition in dysgenic and reciprocal males. And because for technical reasons, it was easy to do even more, so we did it in dysgenic and reciprocal females as well. So what we did is we took these different F1 males that we want to compare, we back-crossed them to the M-type parent, which lacks P elements, so there's no P element signal coming from the M-type parents. And in that F1, any transpositions that happen in their germline will show up in their offspring. So we're looking for new P element insertions in the offspring of these F1s. Right. So we did this on the cheap by using targeted sequencing. So we did this really complicated library prep, and it allowed us to screen over 2,000 flies. So we used targeted sequencing. We used, we did pools of 8 flies. So we're hoping that we can identify new transpositions in each pool of 8 flies. We had a molecule that helped us identify the pool. We had a molecule that helped us identify the we pulled these pools of 8 flies into larger pools, so we didn't have to pay for, we ended up preparing 12 libraries in the end for 2,000 flies. And we also had unique molecular identifiers to identify, to prevent us from counting the same molecule twice. This is a PCR-based assay, and you can have PCR duplicates, and we didn't want to count PCR duplicates as independent evidence of transposition. So we just used UMIs to get rid of these PCR duplicates, unique molecular identifiers. Right, and we used a TE-specific primer and ligated, chopped up the genome, used a TE-specific primer to amplify the TE and whatever flanking sequence was nearby. We used that flanking sequence to identify the location of the TE. So we're looking for new locations which suggest the TE has just arrived there. And we did this for the P element, and we also did it as a control for a different transposable element called Ru. Okay, so what we see here is on the left, we have the P element data. You can pretty much ignore everything but the blue and the red. So the blue are the reciprocal controls, which should have well-suppressed P element. The red are the dysgenic flies. And we count, each dot represents a unique insertion in a fly. So by unique, I mean it's an insertion unique to a pool that's at least 1 kb away from any other P element indicated. You can see there's, I've lost my statistics. However, there's a lot more P element insertions in the dysgenic cross than there is in the reciprocal crosses. And because this was quite a complicated, weird thing we did, I wanted to have a control. You can see that Ru, there are many more copies of Ru in the genome than there are of P element, but there's nothing really going on with respect to transposition in either the dysgenic or reciprocal crosses. So we measured the transposition rate as around, whatever that is, something on the order of 10 to the minus 3 to 10 to the minus 2 per copy per individual. So this is similar to indirect measurements, direct measurements in Drosophila melanogaster, and a bit lower than those that Robert estimated in a cage by inferring. So we found that we compared males and females. As I explained already, we had to do these, use this funny attached X-strop to get genetically identical males. We had to do a different cross to get genetically identical females. So our males and females are not quite comparable really, because they're on different genetic backgrounds. So we did see 30% more transpositions in females than males, but we don't know why. We don't know if that's sex or if that's genetic background. Again, there was this suggestion that P element shows a preference for origins of replication. We found that indeed we saw more transpositions into the origins of replication. So we have, presumably that's got to be an insertion bias. Okay, so there is some consequences for this B element dysregulation. There's a higher transposition. What else? So the RNA-seq data I showed you before was just looking at expression differences between dysgenic and control males in their transposable elements. This is their expression differences due to genes. There's a lot more going on. There's a lot of, that looks really crap, but there's a lot of red and orange dots. Those are genes with significantly different expression between dysgenic and reciprocal F1 males. There were almost 2,000 genes with differential expression, at least twofold. Right, so why does this happen? So when we did this RNA-seq, basically what we did is we pulled out testes, smashed up the testes, got their RNA, and looked at the RNA-seq data using the standard DE-seq2 method. However, those testes are made up of a bunch of different cell types, and it could be that some cell types are missing from one of the different kinds of flies, or there's a change in abundance of one cell type versus another. And that could explain why you see lots and lots of difference in gene expression. It's not that each individual gene is regulated differently in dysgenic versus reciprocal flies, but instead that you're just missing a cell type where one kind of gene is expressed or not. So we used some single cell data, or single nucleus data rather, from melanogaster to try to disentangle this. So this is data from the Raz et al. Study. And they characterize the genes that tend to be expressed in one cell versus another. And we compared those to our up and down regulated genes to look for cells where our down regulated genes are preferentially expressed, and we assume that those cells are missing, or we can infer that maybe those cells are missing. And cells where genes are in our upregulated data set are expressed more might be overrepresented in our data set. Okay, I hope that wasn't too confusing. I feel like it's simple in my head, but it was confusing when it came out of my mouth. So we looked at what cell types were enriched or de-enriched in dysgenic testes. And what we see here is on the top, you've got your germ cells. So these are the sperm as they're developing, starting with the initial stem cells and going on towards mature sperm cells. You've got the cyst cells, which are kind of somatic cells associated with sperm cells, again, going on through development from top to bottom. And then you have other somatic cells that are associated with testes, like muscles and I don't know what else. If you look at the top part of the plots where the sperm are, there's a lot of blue. Those are our down-regulated genes. So it appears that the sperm are de-enriched in these testes for dysgenic males. So the dysgenic males do appear to be experiencing germ cell loss, just as the females are. The P-element is murdering their sperm. And then, of course, there's a corresponding increase in the associated somatic cells. Okay. Does this show up in fertility? So we did some fertility assays. They were not very exhaustive, so we didn't try to sperm exhaust the males. We just gave them some females and counted how many offspring occurred every couple of days. So when we do this, we've got dysgenic males here in the red and reciprocal controls in the blue. And you can see that there's not a huge difference between the two. There's maybe some lower peak fertility, which is significantly different, but it might be hard to see the confidence limits. Lower peak fertility for the dysgenic flies, but eventually they catch up and they have similar numbers of offspring. One thing that is different is we see more, about 10% sterility in the dysgenic flies and none in the reciprocal flies. And that's a significant difference. I seem to have lost my stats again. So there is more sterility in these dysgenic flies, but overall, compared to females, the effects are very mild. So when we do these same sorts of crosses and look at female fertility, the dysgenic females here in red and the others, the controls in gray. This is data from that was collected here by Tom Hill. It was actually supposed to be my experiment, and then I kept going out of town, so he had to collect the data anyway. You see many more sterile females, a sterility rate of about 50%, compared to about 10% for males. So females are about 50%, sorry, right. Females are about 50% sterile, and this is at 25, which is usually in a temperature where dysgenic effects are more mild. You also see lower rates of offspring overall, even for those females that are fertile. They just don't have as many kids. It is about 5 times the rate of sterility and about half the number of offspring, if I remember, that females have, even at 25 when dysgenic effects are milder. So males show milder fertility defects compared to females. Yeah. So just because we had the data, we decided to look at how P elements are regulated in females, in males. And so we wanted to know about things like piRNA pathways. Are they operating in testes? This is not the first time it's been looked at, but we wanted to know how they're regulated. They're regulated by these piRNAs, which I mentioned a few times. They're small RNAs. The P-type stocks have these small RNAs that match the P element. And that's important because the way they suppress the P element is by homology with it. So you have piRNAs that match the P element and target that mRNA for degradation. When that mRNA is targeted for degradation, it ends up being chopped up, and it ends up being chopped up in this very particular pattern where there's overlap between the two different strands of RNAs. Right. So some of this negative regulation that the piRNAs do is post-transcriptional. It chops up RNAs and results in a characteristic 10 nucleotide overlap between sense and antisense piRNAs. So when we look in our RNA-seq data for expression of these piRNA genes, we see lots of piRNA genes that are expressed. It appears to be very similar to what goes on in females. There is one kind of exception, which has been suggested before, which is this post-transcriptional regulation. A lot of it is done by these two genes, aubergine and argonauts. And it seems like in testes, aubergine may be doing more of the job, which is probably something only of interest to piRNA nerds, but nevertheless, I thought it was kind of interesting. Right. So we've got these two genes doing the chopping up, and we're looking now for the 10 nucleotide overlap between the two, and which gives us an indication that this post-transcriptional regulation is happening in the male germ-line as it is in the female germ-line. And we can, in fact, see this 10 base pair overlap between sense and antisense RNAs, which suggests that, again, you have this germ-line suppression mechanism acting in males as it does in females. If we look at the P element specifically, we also see this piRNA signal. It's a lot messier because you're only looking at a very small subset of the data, and also you're looking at flies that don't have very many P element copies. But you see enough of a signal to convince us that the P element is also regulated by ping pong in the testes. There is another way which P elements can be regulated. In particular, this is well known from classic work, and that is by alternative splicing. So most P element transcripts, for most P element transcripts, okay, start over. The P element has 3 introns, 4 exons. The exons here are in blue, the introns are in orange. Those introns are spliced out need to be spliced out in order to make a functional transposase protein. If you look at what happens for most transcripts, you only have splicing of the first 2 introns, and the 3rd intron remains. This results in a truncated protein, and you end up with inactive P element if you keep that 3rd intron. If you splice it out, then you can have active transposase, and that's what you need for P element activity. So there is some suggestion that this is the main way in which P elements are regulated, that they're regulated by retaining this 3rd intron so that you suppress functional transposases. And if you have dysgenic flies, what happens is you disrupt that splicing suppression, end up with spliced transcript, and end up with functional transposase. Oof. Okay, so we looked at our transcripts, and we categorized these into these different categories. So we counted roughly 829 transcripts from dysgenic flies and 166 transcripts from across our reciprocal samples that show retention of this third intron. So this is cases where the P-element is suppressed. Obviously, we have more transcripts in the dysgenic flies than we do in the reciprocal flies because, of course, there's more P-element expression in the dysgenic flies. But also, proportionately, those transcripts are more often spliced. And when you do the stats, it's significant. We have 32 spliced transcripts across all our dysgenic samples and none in the reciprocal samples, even though if they were spliced at the same rate, we'd expect, I don't know, 10 or something. Okay, so we've got splicing suppression. What else do we have? Right. Or splicing suppression disrupted in dysgenic flies. What we don't see are more piRNAs that match the P-element in the dysgenic versus the reciprocal flies. So this is differential expression of our piRNAs between dysgenic and reciprocal flies. Nothing's going on in general, and the P-element is unremarkable even by those standards. There's the same number of piRNAs in the dysgenic and the reciprocal flies. It's not like the reciprocal flies have lots more piRNAs, and that is helping suppression. Okay, I will wrap up quickly. So I'm just curious, basically, wanted to see how TE expression and piRNA expression are related generally. So these are piRNAs on the piRNAs on the y-axis, TEs on the x-axis, and this is just transcripts per million for each TE. So the piRNA that matches the TE and the transcript from the TE, and how those two things are correlated. So we correlated these across all of our samples. There's a strong enough positive correlation to be reasonably convincing. And this kind of positive correlation makes some kind of sense because a lot of these piRNAs actually come from the transposable element message being chopped up. So the more TE mRNA you have, the more you have to chop up and to make into piRNAs. So you have positive correlation there. However, if you look for the P-element, you instead have a strong negative correlation. So you've got the more P-element transcript that you have, the fewer piRNAs that you have, relative to your piRNA. Sorry, relative to your. Anyway, it's easier to see on this plot. We'll just look at this plot. You have here. Oh, it's really messed up. However, you can pay attention only to the red dots and the blue dots. So you have, in the red dots, you've got your dysgenic males, and the blue dots, you have your reciprocal males. Up at the top is the transposable element messenger RNA, and down at the bottom is the piRNAs. And the dysgenic flies, you have 8 more or 20-fold more transposable element mRNA than you do piRNAs. And then reciprocal, they're much more similar in level. And you have, which makes sense because you have more suppression of the P-element. Okay, so, and just for fun, I did a principal components plot, just looking at expression in these different types of TEs and expression of piRNAs in these different types of samples. I just wanted to see what this looked like generally and what explains the variation in TE expression and in piRNA expression in these different flies. That looks really terrible. It didn't look great in the original, but somehow it's really messed up here. This is a plot that's meant to show a vector that shows you the biggest expression difference on PC1 and the biggest expression difference on PC2. The biggest expression difference on PC1 is driven by P-element expression, and that makes sense because you have big expression differences in the P element between your dysgenic and reciprocal flies. And that's also true for small RNAs. This is really hard to see. I apologize for that, but the M stalks and the P stalks are here as well. So you have the P element completely absent from the M stalk and strongly expressed in the dysgenic flies, and that's what explains the variation on PC1. If you look at PC2, what explains the variation instead is a transposable element called shell deer, which is also a recent invader of Drosophila simulans. It invaded a little bit before, and it's actually more highly expressed in the M-type stock than the P-type stock. So it's the reverse situation. Oh, nearly done. Okay, so just quickly to summarize, attached X crosses can give you these F1 reciprocal flies with identical nuclear genotypes. And I'd just like to point out, because I had forgotten this until I was writing up this work, that attached X was discovered by the other Morgan. So I showed the picture of Thomas Hunt Morgan at the beginning. This was his wife. So she did this, found this really weird phenomenon. And I really recommend reading the paper. It's called Non-Kristoff Inheritance and something or other. It's just really fun to read these old style papers with no references where they're just trying to figure things out. And, you know, it's really genius. Dysgenic males in this cross show higher P element expression in testes, higher P element transpositions, but similar levels of pyranase as control males. These dysgenic males show potentially loss of mature sperm cells, as inferred from the associated with single nucleus data, and high levels of sterility, but these fertility effects are mild compared to what you see in females. I'd also point out that the transposition rates are relatively similar in males and females. And so what we kind of wonder is, because the transposition rates are roughly similar on the same order, and the fertility effects are milder, that P elements may be spreading more easily through males than they are through females, because you have jump as much, but you don't kill the ability to pass on genes. I hope that was halfway clear.